There are three types of duplicates. You have the optical duplicates, and they occur when a cluster is falsely called as two clusters. And this happens, for instance, when a cluster is on the border of two tiles. And then it will be called twice, one for each tile. How can you find these optical duplicates? Well, you look at the IDs of the reads, because the IDs contain the location on the flow cell. So if you find two reads with very close locations on the flow cell, they are probably optical duplicates. And then you have the library duplicates, and these are the ones that are generated by PCR during the library prep. How can you detect library duplicates? Well, they will have the same sequence. So they are detected by looking for reads that have exactly the same sequence. And then third type of duplicates are the clustering duplicates, and they occur mainly on patterned flow cells. So traditionally, Illumina sequencing is done in these flow cells here, so the glass slide with the metal grid on them. But in newer sequences, these patterned flow cells are being used that have these tiny wells. So what happens? Well, if you have a fragment of your library that ends up in this well here. So what happens? A fragment of your library ends up in these wells. Yeah, it binds to a primer on the surface of these wells. And then you know that the other end of the fragment will bind to a different primer. And then a bridge is formed, yeah, where one end of the fragment is attached to the surface of the flow cell and the other end of the fragment is binding to a primer on the surface of the flow cell. So the longer your fragments, the longer these bridges will be. So if you have rather long fragments, it can happen in these patterned flow cells that one end of the fragment is attached here in this well, while the other end of the fragment is binding to a primer on the surface of a neighboring well. And then a bridge is formed between these two wells, and as a result, these two wells will represent the same copy from the library. So these are typically clustering duplicates. How can you find clustering duplicates? Well, again, it will be reads that have exactly the same sequence. So what you're going to do is you're going to use a tool that will remove or mark the duplicates. And again, marking means that you are going to add information to the BOM file that says, hey, this read is a copy of another read in this file. It's not possible to make a distinction between duplicates that arise from PCR amplification and real biological duplicates. They will both be duplicates, they will have the same sequence, so yeah, there you are. And this is why they don't mark duplicates for RNA-seq data. The only type of technical duplicates that you can identify are the optical ones. But for technical replicates that arise from PCR, you have no way to recognize them. For RNA-seq data, you can use these UMIs. So you add one UMI, one unique UMI for every transcript. So if the transcripts are duplicated during the library prep, they will have the same UMI. But in bulk RNA-seq data, these UMIs are not heavily used. So in single cell RNA-seq data, they are always used. In bulk RNA-seq, not that much. But you can use them, and it's a big benefit because it will allow you to make a distinction between the biological and the technical replicates. So of course, if you have them, then you can remove your technical replicates from your RNA-seq data, and then you would have to mark duplicates there also.